Ladies and gentlemen, thank you for standing by and welcome to SARS CoV-2 in Indoor Air Principles and Scenarios. At this time, all participants are in a listen-only mode. If you have any questions, you may submit them via the chat function on the webinar. And if you require any further assistance, you may press *0. Thank you. I would now like to hand the conference over to your speaker today, Mr. Vito Ilacqua. Sir, the floor is yours.
Welcome everyone. I'm Vito Ilacqua. I'm a scientist in the Indoor Environments Division of the Office of Radiation and Indoor Air at EPA. Over the past months, our team has been monitoring the growing research to protect people from the novel coronavirus in indoor environments and really translate its implication for public health. So as a part of that effort, we are very pleased to have such exceptional speakers today sharing their research on a topic that is affecting so many so directly. In fact, we maxed out with our registrations, but the webinar will be recorded. You should expect to see it perhaps a couple of weeks after today. Today our speakers will present one after the other for the first 45, 50 minutes or so, and then we will have a questions and answers session at the end. Please enter your questions only through the Q&A box on the lower left of your screen, and you can do so throughout the presentation. Because of the size of the audience, we will try our best to combine similar questions, but even so, we may still not be able to get to all of them. Just out of consideration for our speakers, let's try not to put them on the spot with policy-related questions that are outside of their expertise. So actually, on that note, before I introduce the speakers, let me emphasize that the views expressed in this presentation are solely those of the authors and do not necessarily reflect those of the EPA. So our speakers today, let me start with Linsey Marr. While many of us in the indoor air research community have been scrambling really to catch up and learn about aerosols and viral transmission, Dr. Marr has been publishing about both the aerosol science and infectious diseases and their intersection for a number of years. And that's really the exact combination of expertise that we need now. She's also been very active in shaping the thinking of the research community in the past months. And if you've been following this topic at all, you've certainly have read or quoted in the press, and for a good reason. She's currently the Charles P. Lunsford Professor of Civil and Environmental Engineering at Virginia Tech. She'll be immediately followed by Dr. Rich Corsi, and he's really one of the pillars of the indoor air research world, and so I will not attempt to summarize his expansive work or pick from his accomplishments here. He's currently the Dean of the College of Engineering and Computer Science at Portland State University. Also often in the news and extremely busy at the moment, as you can imagine. So again, thank you for offering your time for this. But I will mention that he's the chair of the prestigious Academy of Fellows of the International Society of Indoor Air Quality and Climate, which Dr. Marr has also been inducted into. I say this just to give you a sense of the respect and recognition that our speakers command from their colleagues in the research world. And with that, Linsey, take it away.
Thank you very much for the introduction. I am really pleased to and honored to give a seminar as part of the EPA series. My research has been motivated by EPA's work on air pollution, starting with outdoors, and then I moved kind of indoors in the past 15 years, but really EPA has been a big supporter of our research, and some of my alumni have gone on to work at EPA. I am trying to advance the slide. Let's see. I don't see the presentation.
It's coming up in a minute.
Are you able to go back to the beginning? Thank you. We will be talking about SARS CoV-2 in indoor air. Thank you. Principles and scenarios. I'll talk about the principles, and Dr. Corsi will be talking about scenarios. I'll be covering 4 topics today: transmission routes for infectious disease, of respiratory viruses in particular, the size distributions of droplets and aerosols that we need to be thinking about, and how evaporation affects those. We will look at viruses in droplets and aerosols, and then talk specifically about what we know about SARS coronavirus 2. For respiratory diseases, there are 4 main routes of transmission. These include direct contact, which is shaking hands, for example; indirect contact, where the sick person leaves a pathogen on an object, and the other person touches that object, such as a doorknob, picks it up, and transfers it to their mucous membranes. The third one is by large droplets, which is if you're close to someone and they cough or sneeze in your face, and those large droplets fly through the air following a ballistic trajectory and land in your eyes, nostrils, or mouth, they can transmit disease. And then aerosols, which are just smaller droplets, ones that are small enough to remain floating in the air for minutes to hours, where someone else can breathe them in. Transmission by large droplets is typically traditionally defined as involving droplets larger than 5 microns and happening only at close range, less than 1 or 2 meters. Transmission by aerosols, also called airborne transmission, is defined as involving aerosols, or things that are smaller than 5 microns, and happening mainly at long distances, greater than 2 meters. The origin of this 5 micron cutoff is not clear. As you'll see later, this cutoff is not supported by modern aerosol science, and I think this distinction has hampered our understanding of transmission. So here's what's really important in distinguishing droplets from aerosols in the context of transmission. The distinguishing feature should really be the route of exposure. Whether droplets land on your body, and we call that contact transmission, so these are fairly large, fly through the air directly from the person who's sick, versus inhalation of smaller ones that we call aerosols that then enter the respiratory system. We've heard a lot about how SARS coronavirus 2 is transmitted mainly at close contact, so when people are within 1 or 2 meters of each other. And at close contact, there can be multiple routes of transmission. So this figure shows person A, who is infected and is releasing lots of droplets and aerosols into the air. Person B is in close contact with them. I think the larger droplets are green and the smallest ones are blue. Kind of red are in between. So you can see that person B is kind of right in that plume and may be subject to the spray of large droplets, but is also breathing in aerosols. Meanwhile, person C, who's farther away, isn't exposed at this point. However, later in time, as those aerosols have spread through the air, you can see them kind of advancing to the right, whereas the larger droplets, the green ones, have fallen on the ground. They've settled. The red ones are settling too, but it's really just the smaller ones that remain floating in the air. those begin to reach person C, who then eventually can be exposed to aerosols and can breathe in the virus. And indoors, this is why ventilation is important, because it can help eliminate the buildup of aerosols if you replace that air with outdoor air, which presumably contains no virus. This is also why face coverings are important for, they can offer the wearer protection, but they are also important for source control. So we can see that if we put a face covering over person A, that will greatly reduce the amount of droplets and aerosols that they release into the air. Even if it's not by 100%, any reduction is better than nothing. So we've seen that transmission by inhalation of aerosols can happen both at short range and at long range. So why is size so important? Well, the figure on the left you probably recognize from EPA's material on airborne particulate matter. We're very familiar with PM10 and PM2.5. For reference, a human hair is about 50 to 70 microns in diameter. PM2.5 is 2.5, of course. The virus itself is around 0.12 microns in diameter, which is similar in size to influenza virus and adenovirus, and a bit larger than rhinovirus. So the ones on the bottom, rhinovirus and adenovirus, are causes of a common cold. Influenza causes the flu, and what everyone's interested in right now is SARS coronavirus 2. Another difference between these viruses is that the ones on the top have a lipid envelope, so a kind of a cell, a lipid bilayer derived from the host cell, whereas the ones on the bottom do not, are so-called unenveloped. But the virus, when it's traveling through the air, when it's released from someone's respiratory system, is not naked. So even though the virus may be 0.1 microns in size, it is released in respiratory fluid droplets that range anywhere from roughly 0.2 microns to 100 microns in size, and even up to 1,000 microns, so really a millimeter potentially for those very large droplets, although those rapidly fall to the ground. So the size of these droplets is what determines the lifetime in the atmosphere and where the virus ends up depositing in the respiratory system. So even though the virus itself might be 0.12 microns, we need to think about the size of the carrier droplet or aerosol when trying to predict its fate and transport. The lifetime of these droplets and aerosols can be predicted by estimating their settling velocity, which is a function of their size, and then using that velocity to figure out how long it will take them to settle from a height of, let's say, 1.5 meters. For those of us who are used to thinking in feet and inches, that's 150 centimeters, and I know that I'm 160 centimeters tall. I'm 5'3", so there's your reference point. But the settling velocity can be calculated using the equation shown here in this slide. It's a function of the particle diameter squared, the density, which we'll consider close to that of water, and gravity and the viscosity of air. So if I go ahead and calculate this velocity and then apply it to droplets that are released from a height of 1.5 meters, you can see I get the red line, which shows how long it takes for those droplets to settle. And we can see that ones that are very large, beyond 20 microns or so, settle within a minute or two. But once we go to the smaller sizes, 10 microns and less, those can remain suspended for many minutes. And in fact, if we look at 5 microns, that takes actually over half an hour, over 30 minutes, to settle to the ground from a height of 1.5 meters. So obviously it can travel during this time more than the customary 1 or 2 meters. So these figures show the trajectory of a droplet of different sizes. So each color indicates a droplet of a different size. And the trajectory it would follow if the indoor air velocity were low, 5 centimeters per second on the left, or high, 20 centimeters per second on the right. So let's focus on the figure on the left. So we have this droplet, 5 microns in red, shown released from a height of 1.5 meters. And you can see that during the time it takes it to settle, it can travel over 100 meters, or well across the room. This is not quite, this is a little bit different from reality, because in reality we don't typically have a steady air velocity indoors just in one direction, but it helps give us an idea of how far things can be moving. You can see that a 10 micron droplet can travel over 20 meters away, and it's really not until we get up to 30 microns, the blue line, where we see the droplet actually does settle within just a few meters away. The right figure shows those trajectories now with the x-axis is stretched out because we have a higher indoor air velocity of 20 centimeters per second. Oh, I should also say that so when the droplets are first released from talking or coughing or sneezing, they're carried by the jet that's coming out of the person's nose or mouth. But within a few seconds, that things kind of mix and disperse and slow down, and the velocity of that jet becomes zero, and then the droplets are subject to the currents, to air currents. So humidity is important because it affects both the size of the droplets and the chemistry. And I'm showing here kind of from high to low humidity conditions. The reason I'm going from high to low, from left to right, is that when a droplet is first released from your respiratory tract, it's in essentially a saturated 100% relative humidity condition. And then as it hits ambient air, which is typically less humid, it will undergo evaporation. But let's start on the left by looking at this droplet when it first comes out of your mouth. It contains the virus, and it also contains salts and proteins that make up anywhere from 1 to 9% of the mass of the droplet. Now, at medium relative humidity, a lot, but not all of that water will evaporate, leading to concentration of the salts and proteins, and the virus is still there. These figures are not to scale in that the amount of salt and protein is probably close to 1 million times the mass of the virus. So we wouldn't be able to see the virus in this. And then at very low humidity, the water evaporates completely, leaving behind just a solid particle of residue of the respiratory fluid and the virus. Now, at medium relative humidity, so at low, people often assume that, well, if the droplet dries out completely, then the virus can't survive. But it turns out that enveloped viruses survive very well when desiccated. But at medium relative humidity conditions, viruses like SARS coronavirus 2 and influenza appear to decay faster under these medium humidity conditions, somewhere above about 40% relative humidity, on up to higher humidities, but we like to cut it off at 60% to avoid mold growth indoors. So we've seen that droplets can easily travel more than 2 meters on air currents, and that humidity affects both the size and the chemical composition of the droplets. So the next question is, how many droplets are there, and how big or small are they? There have been many studies on this question going back as far as 1946, so it really shouldn't be a surprise to us that we release droplets and aerosols from coughing and also just from talking. But here's a study by Bill Linsey that was published in 2012, where he looked at the number of droplets emitted from a cough when a person had the flu and after they had recovered. And they looked at droplets in the size range from 0.35 to 10 microns, and you can see that when the person was sick, they released on average 75,000 droplets in one cough. After they had recovered, that number went down, but if you look at the standard deviations, there wasn't a significant difference. And if you look at the size distribution of these, you can see there are a lot of droplets that are smaller than 1 micron. There was a comprehensive study done by Linsey Marr and colleagues in 2011, following up on an earlier study of hers, where they used multiple methods to look at the full range of droplet sizes. So you have to be careful when reading through the literature on this topic, because different groups use different instruments to measure the size distribution. And those instruments have different ranges in terms of what sizes they can detect. So this one was fairly comprehensive because they used both an aerodynamic particle sizer, which can go down to 0.5 microns or even lower, up to 20 microns. And then for the larger droplets, they measured those by droplet deposition analysis. So they put out slides essentially and looked at how the droplets that settled on there and how big they were. The resulting size distribution is shown by the black dashed line. The top figure shows it for speaking, and the bottom figure shows it for coughing. These concentrations are numbers in your exhaled breath, so right as it's exiting your nose and mouth. You can see that the distributions are bimodal. They're about 10 times higher for coughing than for speaking, so the number concentration per volume of air coming out of your mouth is higher with coughing than speaking. And we can see that the first mode has a peak around 1 to 2 microns. So there are plenty of droplets down in this kind of aerosol size range of smaller than 5 or 10 microns that can stay suspended in air for many minutes to hours. And there's a second mode somewhere between 100 and 200 micron. Those will fall out within seconds or so. We've seen that breathing, talking, and coughing can release droplets that range from submicron to millimeter in size. There are other studies with flu virus showing that the virus is found in the aerosols. In fact, there's more of it than we would expect based on just the volume of the droplets, more of it than we expect in the fine aerosols, or the ones smaller than about 5 microns. So now we'll talk about what do we know about SARS coronavirus 2 in droplets and aerosols so far. This is from a study in a hospital in Singapore where they swabbed surfaces and got an air sample. So what we want to know is, well, is the virus found in air, and what size droplets is it found in, or aerosols, and then how well does it survive? So that's what we'll walk through now. And they found that the samples listed in the upper right were positive for the virus. They were trying to detect virus RNA, which tells us, yes, the virus is there. What we don't know is whether it's infectious or not. But you can see that virus was found on the table, the bed rail, the locker, light switches, on the floor, the toilet, and also on the air outlet fan of the room. So the air outlet fans of the room are number 15, indicated with the 2 red pluses over towards the left of the room. They're near the patient's bed. They also found some negative samples with different surfaces. Those are listed in the lower right-hand corner. Their air sample, they took one air sample from one location, and that sample was negative. Now, it's not clear. Well, if you imagine the patient lying in the bed and you look at the location of the air outlets, you might imagine that anything released by the patient is going to immediately exit the room through those outlets. So unless you actually collect the air sample within that path, I think there's a low likelihood that you would detect any virus, especially given that hospital rooms such as these have high ventilation rates with an air exchange rate of usually 12 to 15 per hour. There have been studies where researchers have detected and measured viral RNA in hospitals. One is a study in Nebraska at the National Quarantine Unit and National Biocontainment Unit, and they found average concentrations in air of on the order of 1,000 RNA copies of the virus per cubic meter of air. In another follow-up study in Singapore, they found they collected aerosols and size segregated them at cuts of 1 micron, 4 microns, 1 and 4 microns in 3 different rooms. You can see that they did not detect virus in room 1, but they did detect it in rooms 2 and 3, again at concentrations on the order of 1,000 copies of virus per cubic meter of air. And here we see that nothing was found in the particles smaller than 1 micron. but plenty was found at 1 to 4 microns and also at greater than 4 microns. Based on this information, we can estimate a viral RNA emission rate of 10,000 genome copies per minute in these small droplets, or let's call them aerosols, aerosol particles. A genome copy of a virus, or detecting its RNA, does not mean that the virus is infectious. Typically, when viruses are made in the body, maybe 1% of them might actually be infectious, that is able to cause infection. So this finding of 10,000 genome copies per minute, which sounds like a huge emission rate, maybe only 1 to 100 of those are infectious. The next question, okay, so virus has been found in air in particles small enough to remain suspended for a while. The next question is, how well does the virus survive in aerosols? And there was a study that came out a couple of months ago where they looked at survival of SARS coronavirus 2 compared against SARS coronavirus 1, which caused the original SARS from 2002 and 3. And they found a half-life of the virus of 1.1 hours. And these were in aerosols generated from culture medium at a humidity of 65%. So the virus can survive for an hour or more. The figures on the bottom show virus survival on different types of surfaces, if you want to check those out later. But contaminated surfaces are not thought to be a main route of transmission of the disease. And then finally, in the last couple of weeks, there was another study published on survival of the virus in aerosols, and you can see that it survives quite well. The left-hand side shows PFU, which is plaque-forming units, or a measure of infectious virus, and it remains fairly stable all the way out to 16 hours. So this is really the longest I've seen for a virus to survive in aerosols and may contribute to the rapid spread of the disease. There are challenges, so some people say, well, you haven't found infectious virus in air yet, so it's not happening. There are sampling challenges, of course. There are low concentrations of RNA, viral RNA in air, maybe 1,000 copies per cubic meter, as we saw in those hospital studies. We might need anywhere from 100 to 10,000 copies to get one infectious virus. I've heard from friends who are virologists that it might be closer to 10 to the 4th for this virus, which means that you would need to sample 10 cubic meters of air. In previous studies, they've mostly sampled around 1 cubic meter of air. Now, ideally, you want size separation. So here's some of the ways to sample and some of the limitations of these. So on the lower left-hand side, I show pictures of the Cascade Impactor and the NIOSH Biosampler, which do offer size separation, but they have fairly low flow rates, which would require 20 to 40 hours of sampling. The middle figure shows more of a high-volume sampler, but the filter that collects the particles or aerosols from the air is kind of the white circle on the end of that instrument, at towards the right-hand side. That's a pretty large filter, and so to elute it, you have to put it into a large amount of liquid, and then there's the challenge of trying to concentrate that, or maybe you just use a subset of it, but then you're throwing away a lot of the virus. And then when you're sampling in air, there can be loss of infectivity of the virus. It can fall apart, as shown here, during sampling. So although we haven't seen detection of infectious virus yet in air, I think epidemiologically, based on what we've seen for transmission, it is happening, and based on what we have seen about how masks work. So I want to end by talking about the 3 C's in masks. So the World Health Organization last week recognized the possibility of transmission of SARS coronavirus 2 via inhalation of aerosols, or airborne transmission. And the way to mitigate that is to avoid what they call the 3 C's, which they took from Japan, which is, first of all, avoiding crowded places with a lot of people, because the more people you have there, the greater the chances that there will be someone who's infected. Second, avoiding close contact settings. So again, transmission seems to occur largely at close contact when you're within 1 to 2 meters of someone for an extended period of time, maybe 15 minutes, maybe half an hour. And then also to avoid closed, confined, and enclosed spaces that have poor ventilation, because virus can build up in the air there. And even if you're not close to someone, you could then be exposed. And then finally, I want to emphasize that there's more and more evidence showing that masks are effective. The idea of masks is both for source control and personal protection. This diagram shows that if the person who's infected is releasing a lot of droplets and aerosols into the air, putting a mask on them, as shown in the bottom, will greatly reduce that and then greatly reduce other people's exposure. Finally, I'd like to acknowledge all my collaborators and students and postdocs who worked on this, along with NSF, EPA, the Sloan Foundation, and some NIH and some other centers that I'm a part of that have contributed to this research. Thank you very much.
And thank you very much, Linsey. And we'll see now to transition to Rich in the meantime. Rich, whenever you would like.
So thank you, Vito, and thank you, whoops, if you can go back to the very beginning, yeah. We're into my extra slides now. All the way back. So while we're clicking back the slides, let me say thank you to Vito and thank you to the Indoor Air Quality Group at the U.S. Environmental Protection Agency for all that you do to educate the public. And like Linsey, the U.S. EPA has funded a lot of my research over the years, including a recent 4-year study of indoor air quality in high schools that I will allude to today. And I also wanted to thank Linsey for that wonderful presentation, because she's defined many things that I don't have to define, so I can get into sort of the practical scenarios associated with exposure to and dose of SARS CoV-2. And my presentation is entitled Aerosols as Vehicles for Dose of SARS CoV-2, particularly dose in the respiratory system. My motivation for studying indoor air quality is defined by this slide, and I hope that it's a reason why many of you are interested in indoor air quality. Americans are amazing indoor creatures. On average, we live to be 79 years old. We will spend 69 of our 79 years of life domiciled inside of buildings. We spend a greater fraction of our lives inside of buildings than whales spend completely submerged below the surface of the ocean. So think about that. We spend 54 of our 79 years of life domiciled inside of our homes, 6 years outdoors, and 4 years in motor vehicles. And the plot that you see right below those numbers is basically the average American day. This is, I think, about 10,000 respondents where they spend time during the day. And the different areas that you see on this plot, A is residence indoors. So you can see how that changes as a function of time during the day. But it's the huge swath of the American day. B is inside of a motor vehicle, which is actually quite a large chunk of the plot. C is in the workplace. D is in public buildings or schools, and schools are a big chunk of D. E is bars or restaurants. And then F is other indoor environments. And I'm going to try to touch on some of these other indoor environments as well as the big ones today in my presentation. So as Linsey said, particles are essentially vehicles for transport of SARS-CoV-2, and they're also, therefore, vehicles for the dose of SARS-CoV-2 in our respiratory system. We inhale tiny droplets when we're near an infector in close contact, so concentrated sort of aerosol stream that we're breathing. But even if we're away from the infector in what I call the far field, you can be exposed to aerosols. And I think what's really important to recognize is that in a busy indoor environment, if there's an infector, there's going to be more people in the far field than there are going to be close to the infector. And so there's greater potential for larger numbers of people inhaling viruses that are associated with aerosols that are accumulating in the indoor environment. And also quite often, people spend more time away from an infector than close to the infector. So I think it's important that we try to understand what's happening in the far field. And so I'm going to focus on the far field today. And in particular, I'm going to focus on the far field and the volume of particles that deposit in our respiratory system in the far field. To do this, I've defined what I call an emission to inhalation dose model, EID. This is a screening model that's really intended to sort of for airborne approximations and scenario comparisons. I don't want to get into all the meat and details of the model. For those of you that are mathematically inclined, it's basically the solution of an ordinary differential equation. And the solution is for the concentration or the levels of particles in air in a given size range I, and how they function, how they vary as a function of time, and their dependency upon the emission rate of an infector. And I've included coughing, speaking, and breathing in my model, but that can be expanded, and the frequency of coughing and the fraction of time speaking and, of course, breathing. And then also a number of removal terms, including ventilation, surface deposition, engineering controls, respiratory deposition, and if we want to track viruses on the particles, we can also include an inactivation time or an inactivation rate for time in air. All right, so I'm going to go through the last three of these removal terms very quickly and then start looking at scenarios. So the first one is deposition to surfaces. Right, so this plot shows on the vertical axis the deposition rate to surfaces, and on the horizontal axis particle diameter. I'm going to be focusing on particles that are kind of in the 0.5 micron to 10 micron range, and that range is shown by the vertical dashed lines in this plot. And you can see that in this range, the deposition rate for particles to indoor surfaces for 0.5 to 10 micron particles ranges from about 0.1 to 7 per hour. And at some context, typical air exchange rates for buildings range from, roughly 0.3 to 4 per hour. Some buildings a little less, some buildings more. Depends on the function or type of the building. But the main point here is that even deposition of tiny particles you can't see, to surfaces can start to rival removal by air exchange. So deposition is important. And even small or tiny droplets can contaminate surfaces over time. In terms of engineering control, I consider increasing outdoor air supply and engineering control because it has to be done right. If we're looking at air intake systems and controlling air intake in buildings. So that's lambda in this beta term, the removal term. That's outdoor air exchange. We can also have filtration either by portable air cleaners or by centralized or room-specific filtration. In these particular cases, what's important is this product, which is the single-pass removal fraction of particles in size I. What fraction of particles do we remove in the filter or in the portable air cleaner every time air moves through that or a particle moves through that, multiplied by the volumetric flow rate through the system, either through the mechanical system, what we're talking about, filters in the mechanical system, or through the portable air cleaner. Increasing interest in ultraviolet germicidal irradiation to inactivate or destroy viruses. We can do the same thing here. We can have sort of a single pass inactivation fraction, and then the number of passes depends upon the flow rate through the system. I've just shown here a plot that shows for portable air cleaners this product of single pass removal efficiency times flow rate, which is referred to as a clean air delivery rate. And if you purchase a HEPA-based portable air cleaner, for example, you should see something called a CADR, which is this product in standard cubic feet per minute. You should look at the tag, look at the information, and aim for something on the order of 300 standard cubic feet per minute or more. Right. And lastly, deposition in the respiratory system. This might surprise some people, but if you pack enough people into a poorly ventilated space, the respiratory systems collectively of those people are going to remove a certain fraction of particles from the air. It's bad for them. We don't want those particles depositing in the respiratory system, but it actually changes the concentration in air. And so we can actually model deposition in the respiratory system using a number of different types of models, but basically what we're looking at is deposition in the head region, the tracheobronchial region, or the lowest recesses of the respiratory system, the alveolar region. And the plot that I show here shows the fraction of particles that deposit on the vertical axis versus particle size on the horizontal axis. Again, between these two vertical lines is sort of where I'm focusing. And each of these curves on this plot shows total respiratory deposition on the top, respiratory deposition in the head region, is the second curve, and then down below you can see the alveolar region, and at the lowest, the tracheobronchial region. All right, so you can see that in the range of particles we're interested in, as the particle size goes up, the amount of total deposition in the respiratory system goes up, and there's some variation as you get to higher particle diameters. You get a lower amount getting into the deepest recesses of the lungs and the tracheobronchial region. What's very important, and I think we forget this sometimes, is that just because you inhale air that has particles in it, those particles don't necessarily deposit in the respiratory system. So if we take a 1 micron particle and we just look right up here to the total deposition in the respiratory system, for a 1 micron particle, for this situation, which is nose breathers during light exercise, you get about 45% depositing in the respiratory system, which means you exhale 55% of the particles that you had inhaled initially. All right, they don't deposit. So this kind of model is built into the EID model that I mentioned. I'm ultimately interested in deposited inhalation dose, and deposited inhalation dose is equal to the concentration of particles of a given size in the air, the levels of particles per volume of air, number per liter or number per cubic meter, multiplied by the breathing rate of an individual, which can change by a factor of 20 if you're a couch potato and you're just resting on your couch versus working out on an elliptical machine in the gym, multiplied by the time of exposure, multiplied by the fraction of particles in this size range that deposit in your respiratory system. So that's what inhaled dose, deposited dose is. I think the location of deposition is likely to be very important. We don't have a lot of information yet on sort of hotspots for infection, but I think it's probably important. The model I developed takes the deposited particles and the particle sizes and converts them to a volume of particles deposited in picoliters, or 10 to the minus 12 liters in the respiratory system. You can take that information and you can use it with viral loads from individuals that are infected. So if somebody has a viral load of 10 to the 11th per mil, which is 10 to the 14th per liter, and 1% of those viruses are infectious, that's 10 to the 12th infectious viruses per liter. We can multiply that by the volume of particles in the respiratory system to get the number of infectious viruses, and I give you an example here. Ultimately, we would love to have a dose-response relationship that says if this volume or this many virions or infectious viruses deposit in this part of the lungs, this is the chance of infection, but we don't have that yet. So these are just example outputs from the model. Before I get to scenarios, I don't want to dwell on these too much. This is for a classroom environment actually on my university campus that shows the concentration of particles in the air if the teacher is the infector with a certain cough, speak, breathe regime. And you can see the concentrations of particles of different sizes after the infector comes into the classroom. This curve on the top is the total number of particles in air in this case 0.5 to 4 micron range. What you see on the right is the total particles in air as a function of ventilation. So the orange curve right here is ASHRAE 62.1 2019 ventilation, and the top one is an underventilated classroom, and then we have 2 times the ventilation rate from ASHRAE 62.1. So you can basically see how concentration levels vary as a function of ventilation. In the bottom left C is the number of or the percentage of particles in different pathways. So blue is the fraction of particles or the percentage of particles that are exhausted to outdoor air from the classroom or still in the classroom air when the infector leaves. The orange portion is the fraction or the percentage of particles, the percentage of particles that deposit on classroom surfaces. So you can see at 1/2 of ASHRAE standard, we got 16% of the respiratory particles that came out of the infector up to 4 microns deposit on surfaces. And the top numbers here, that I'm not sure if you can see these or not, are the percentage of particles that deposit in the collective respiratory system of students in the classroom, so up to 3.5%. And on the right here, you can see if we use a portable air cleaner that about 31% of the particles that came out of the infector during a 75-minute lecture end up in the portable air cleaner. And finally, what I'm aiming for is plot D here, which is the number of deposited particles in different parts of the respiratory system, in this case for these different scenarios in ventilation and use of a portable air. So the blue is the number of that deposit in the deepest recesses of the lung, the alveolar region. The orange is in the tracheobronchial region, and the gray is The gray is in the head region, and this is for nose breathers. And the little numbers you see on top of the bars are the actual volume in picoliters that those particles make up. So that's the kind of output you can get from the model. So now I'm going to pivot, and I'm going to pivot to applying the model to an indoor environment where we have really good data, where there was an outbreak of COVID-19, probably the best characterized environment that's been in the published literature on outbreak of COVID-19, and that's called Restaurant X in Guangzhou, China. I won't get into all the details, but there was an intense amount of metadata collected for this restaurant, so we know a lot of information. Researchers were able to go in after the event and do forensics analysis, and so it's great for modeling purposes. And I used the metadata from this restaurant and tracer studies that were done by researchers, and I took the index case, which is an individual on the sort of far side where you see that what looks like a cloud on the far side of the restaurant. The index case that was known to have symptoms and that infected 10 other people in the restaurant. I made assumptions related to the emissions from that infector in terms of coughing, speaking, and breathing. And I only looked at a particle size distribution in this case for 0.5 to 4 microns, so we can certainly extend that if we want to easily in the model. So I did this analysis using the EID model for this restaurant and looked at the volume of 0.5 to 4 micron particles deposited in the patrons that got sick, and it came up with an estimate of 1 to 10 picoliters for particles in that size range for 75-minute exposure when the infector was present in the restaurant. So the range depends upon what I assume about the emission conditions largely from the index case, but also to some extent on what I assume about mixing to the other parts of the restaurant, et cetera. So what I then did is took that information and I said, can I pivot with this? And can I develop a new screening approach where I take the index case in this restaurant, the infected person, and I place them in different environments? What if that index case is your rideshare driver? What if that index case is a staff member in the gym that you go to? What if that index case is a teacher in a classroom? And what I do when I take that index case and put them in a different environment is I calculate a parameter called omega, which is the volume of deposited particles in this new environment relative to or divided by the volume of deposited particles in restaurant X. So what I want is omega to be as small as possible if I put that type of an infector in a gym or a classroom. In fact, I'd like it to be less than 0.1, 1/10 or so of the Guangzhou restaurant case. And so that's what I'm going to do for you right now and show you some results for different scenarios. And I'm going to start with a busy restaurant that has a few less people than was in the restaurant in Guangzhou, China. It's got much better ventilation on the order of about 7 times the air exchange rate than will be found in Guangzhou, China. And patrons in the restaurant are breathing air that's been infected by index case X. And when I do that, these are the curves I get for omega for a restaurant ventilated to ASHRAE 62.1 standards in theory, all right? And then for an under-ventilated restaurant, and then for a restaurant that has significant ventilation, twice the ASHRAE 62.1 standard. So you can see in an under-ventilated restaurant, I'm getting an omega that's above 0.25. I don't like that. I want it to be lower than that. For ASHRAE standard, about 0.17. And then for a well-ventilated restaurant, I can actually get down below 0.1 in a very well-ventilated restaurant. Again, this is for the aerosol route. I'm not considering close contact and fomites, which are also important. I think important points here for restaurants is wear a mask when you're not consuming the food. Distance as much as possible. We should have very well ventilated restaurants. It's safest if you're eating outdoors, physically distanced. Reduce your time. Don't sit around and chat after your meal. Wait outside before your meal. Takeout and delivery. So restaurants can be problematic environments, but we can reduce omega significantly. I also did a choir practice, and with 50 participants in a 1,000 square foot room, 100 square meter room. I assumed that they were singing 50% of the time. I did elevated speaking with a multiplier to get sort of emissions during the choir practice, and heavier breathing since people inhale to a greater extent when they're singing. And again, use sort of standard ventilation in this choir practice. And what I get for this is looks horrible. So omega is on the vertical axis, and I find an omega of close to 1 if the building is underventilated to half of what it would be in theory from ASHRAE 62.1 2019. Even with ASHRAE standard ventilation, I'm still a very high omega. Basically, I can't do anything to get the levels down to an omega value that I want. So I have concerns across the board for indoor choir practices. I know researchers at CU Boulder, Shelley Miller, and others are doing some great research right now on actually characterizing particle emissions from choir practices. It's just not an environment that I think people want to spend a lot of time in right now, and I'd suggest if choirs had to meet, they meet outdoors and distance and practice outdoors. The next one is gym with aerobic activity, where there's 40 patrons. Unfortunately, index case X is a staff member at the gym, and we have people working out on treadmills and ellipticals, doing aerobic exercises with very high breathing rates, which is going to expose them to more tiny droplets that come out of index X's respiratory system. So for a gym, I end up with even worse conditions than choir practice for somebody who's working out aerobically. So you can see omega values that are almost twice what you found in restaurant X in Guangzhou, China. And that's with a poorly ventilated gym, but even a standard, ASHRAE 62.1 2019 ventilation, way too high omega factor. And I can do all sorts of things, and I just can't get the omega factor down to where I feel comfortable going to the gym. I tend to be a gym rat myself. I haven't been going to the gym for months, and I don't plan on going back until there's a vaccine. So from my perspective, avoid indoor gyms. Perhaps if you have a gym that has these huge doors on either side with huge cross flows where you can turn the indoor environment into the outdoor environment, it'll be safer. Do outdoor workouts. If you have to be in a gym, even if it's extremely well ventilated, wear a mask if you're not, if you're able to, and then physical distancing. Ride share. So unfortunately, you call a ride share and index X is your driver. And you get into the cab and you take a 20-minute drive across town at 20 miles per hour, which affects the air exchange rate. These are the results you get in that case. An omega value of 3. So extremely high if you're in this close environment with the windows closed in a ride share. If you crack the windows open just a little bit, you can drop that all the way down to just above 0.5, which is still not low enough for me. This is without wearing a mask. If you wear a mask, you can, and your driver wears a mask, you drop those bars even more. Insist that your driver wears a mask. You should wear a mask. You should crack the windows open. You have control over that environment. Do it. Avoid long trips and busy commutes. If you do all those things, you can actually get omega down to 0.1, which is where I'm aiming to get with these simulations. Elevators. There's been a lot of interest in elevators. So you happen to walk into an elevator and there's index X waiting for you, and you and index X, this infected individual, travel in an elevator for 1 minute without the doors opening. I've learned recently that the elevators are extremely well ventilated, up to 60 air changes per hour, 1 per minute. That helps a lot. So when you actually look at your exposure in an elevator to background aerosols, the omega value is extremely low, much lower than anything I've just shown you. And in fact, if nobody speaks on the elevator, you're looking at less than 1/1000th of an omega value relative to restaurant X in Guangzhou, China. So with respect to sort of airborne contaminants in elevators, I think that the risk is negligible. So the focus probably should be on close contact and fomites, where you basically require that people wear masks on an elevator, have no speaking etiquette, try to reduce the density if you can, and distance and face away. But the airborne aerosol route should be relatively insignificant on elevators because they're well ventilated in such a short exposure time. Did a school classroom where the teacher's infected and is speaking a lot, and the teacher now is index X from the restaurant in Guangzhou, China. And then a student is infected but not showing symptoms, and they're speaking 10% of the time and not coughing. And so for this particular condition, you can see omega values for an underventilated classroom that are quite high. I don't want to be in this classroom. I don't want my children to be in this classroom. I use 1/3 of ASHRAE standard here because I've tested a lot of classrooms in Texas, probably 200 classrooms, K through 12, and I find some that are so underventilated that it's probably less than 1/3 of ASHRAE standard. So this is not an unrealistic condition in some classrooms. ASHRAE, ventilating in theory to ash rate 62.1, still too high for me. I can do a lot of things. I can use portable air cleaners. I can use, MERV 13 filtration with 3x passes, and I'm still having a hard time getting down to 0.1 omega in this classroom. If the student is the infector and not coughing, the numbers are considerably lower than if the teacher who's speaking all of the time is the infector. I think for school classrooms, remember that everybody wearing a mask will decrease these bars significantly, maybe by 50% or 75% reduction. And there are things we can do to continue to try to reduce the value of omega. So I'm going to end with return to school, if I may, in the next 3 or 4 minutes. I think this is on a lot of people's minds, and we'll pivot off of this classroom example. And you can see from the classroom example that there are conditions that can be really bad in a school classroom relative to a case in Guangzhou, China, in a restaurant where a lot of people got infected. So there will be a return to school in the fall in some areas in the United States. I have concerns about many classrooms being underventilated, is my experience, mostly because school districts are worrying about energy consumption and the cost of energy consumption. Many K through 12 schools are poorly resourced. The time to properly prepare for the fall is very short at this point, so hopefully the school districts have started 2 months ago. If they haven't, I have some concerns. And I think there's non-scientific decisions being made right now related to how to go about this in some school districts. I'm not going to mention any examples, but I think it's important to use proven, effective, independently tested technologies. I show some examples here just from schools that I've worked with in Texas. Here we have a downspout off of a roof that goes into a grate that goes into the crawl space of a building that's badly contaminated with mold, with water wicking up the walls. Schools have sometimes bizarre construction, and the facilities and maintenance people, school district doesn't have the ability to make changes that are needed. This is actually an HVAC filter in an affluent school district in Texas. You can see this significant dust cake on the filter and some sort of fungi growing. You can see the white growing on the filter, and here you just have a poorly seated filter with significant bypass. These are all images from schools, and I've got a whole library of these kinds of things. It's tough to do things right in schools for a lot of reasons, and one of them is resources. So for return to school, there's a lot of things we need to do. I don't want to list them here. This is a partial list, but certainly physical distancing. I feel that we have to require masks unequivocally. Masks should be worn in the classroom. We can talk about increased outdoor air supply, filtration, portable HEPA filters, UVGI, and lots of other things, including possibly removing carpet. And so I want to talk about that very briefly. We just finished a project funded by the US EPA, my colleagues at the University of Texas and I. And what we saw in classrooms was a 4-year study with lots of indoor air quality parameters. This is a course particular, or PM10 concentrations, mass concentrations for 3 days in one classroom, but it's a very typical result. And you can see each day you see these huge spikes in PM10 concentration during the day, day 1, day 2, day 3. And each of those spikes corresponds to students coming into the classroom and students leaving the classroom. And you can see an inset at the upper left of day 1. This is a substantial amount of particulate matter that's resuspended primarily from the floor, but perhaps even shedding from students' clothing and that type of thing. So it begs the question, I think, can enough infectious viruses accumulate on flooring, accounting for the rate of inactivation, for resuspension to be relevant? And this is a question I think we don't have an answer to, that we need to find answers to. The precautionary principle tells me that perhaps we need to think about the flooring in schools before school starts. What we found in our study, whether it was PM1, PM2.5, or PM10, is that the average mass concentration in classrooms that have carpet is significantly higher, especially for PM2.5 and PM10, than it is in classrooms that have vinyl tile. It's a lot easier to clean. And so, one question is, should we be removing carpet from classrooms before we start the fall term for this reason? And then just to wrap up, without getting into the details, there's a second model I've been using that was developed by Don Milton's group about 10 years ago that predicts the probability of infection in an indoor environment if there's an infector in that environment. I re-derived the model slightly to put engineering control term phi in the exponential term here. Key to this model is this value Q, which stands for a quanta generation rate. think about that as sort of the amount of infectious material coming out of somebody's respiratory system who's the infector, right? So I applied this model to the restaurant in Guangzhou, China, and I was able to back calculate quanta generation rates for index case X of between 60 to 185 per hour. And so what I'm doing for my own university is I'm varying classroom conditions for over 100 classrooms now. I'm trying to find the time to finish all the classrooms to determine what the maximum capacity or how many students I can have in a classroom to keep the number of disease cases or the number of people that get infected during one classroom period to less than 1. That's my goal, all right? Just like keeping the omega value previously at less than 0.1. And I want to compare this with the maximum capacity for physical distancing in a classroom. And this slide, which is my last slide before my closure slide, is I think a very practical slide that summarizes these results. So the vertical axis shows the acceptable fractional capacity of the classroom. 1 meaning if there's 24 chairs in the classroom, we can fit 24 people in the classroom. 0.5 means you have to operate at half capacity. And the horizontal axis is the quanta generation rate, varied over that range that I showed you for index case X. And I did this for a 75-minute lecture for universities that are on the semester system. This is a Tuesday, Thursday class. And then for a 3-hour time period, for example, if this was a high school or even a middle school where students are spending more time in the classroom. You can see all the parameters for the classroom here. At my university, we have a fairly high percentage of outdoor air in our supply air to each classroom, simply because of the climate that we're in. It's going to be different, say, in Houston, Texas. And so in this particular case, the teacher is the infector, and I treat the classroom either as is with 100% outdoor air, that's curve 2, as is curve 1, 100% outdoor air is curve 2, and then basically a classroom with a portable air cleaner with a HEPA filter with a clean air delivery rate of 300 standard cubic feet per minute is curve 3. And I do this for each, the 75-minute class and the 3-hour class. And so what you can see in here is interesting. If we just look at physical distancing, keeping 6 feet apart, the average capacity in classrooms on our campus is about 0.3. If you have 24 seats in a classroom, you should put 8 people in there if they're all wearing 3-foot radius intertubes when they walk into the classroom to keep 6 feet apart from one another. So you can see that for aerosols, when we compare it to physical distancing, for a 75-minute lecture, if I have 100% outdoor air or a good portable air cleaner, the maximum acceptable capacity is larger than the physical distancing. So physical distancing in this case dominates the number of students I can have in the classroom. If it's a very infectious teacher and I don't do anything in the classroom, I might be okay until I get to about 135 per hour quanta generation rate, after which aerosols take over and I have to drop below that 30% capacity. For a 3-hour classroom environment, it's difficult. Aerosols tend to dominate unless you're at a fairly low quantity for the quanta generation rate. And so in this particular case, just keeping 6-foot distance isn't going to be good enough for a long class period. I think these are practical results. I'm doing this for like as many classrooms on my campus as I can do so that we can act appropriately in terms of controls in the classroom. Remember that these are all done for people not wearing masks. At my university, everybody will have to wear a mask in any building when we come back in the fall, and masks bump these curves upward, or if you want to think of it this way, provide additional safety factor if you operate off of these curves. So in closure, I would say know your environment. As I've just shown you with the simulation, not every environment is created equally in terms of risk. There's a misspelling here. I don't know what hot or means, but take great caution in environments that are poorly ventilated, that are confined spaces, like in an automobile, where there are potential for high emissions, like the water practice, that have high breathing rates if you're the receptor, like in a gym. Our weapons to lower risk are wear masks, have everybody wear masks, as Linsey said, physically distance, ventilate, go outdoors. Any activities you can do outdoors should be done outdoors. Reduce your time in the environment, and then employ engineering controls. And I want to end with a positive note, and that is, for many environments, it is possible in school environments to dramatically reduce risks with the right strategy in many cases. And I think this is critical for return to school. As I showed you, in some environments, we can go to great lengths and I'm still concerned about being in that environment like a choir practice or a gym if I'm doing aerobic activity. That's it. Thank you so much.
Thank you very much, Rich, and thank you, Lindsay. We went a little beyond the original timing, but this was such useful and interesting information, and we'll now begin our question and answer session, and my colleagues have been working to try and make the most of the many questions we have received, and so I'm going to take the first set of questions really to say that yes, the presentation is being recorded and will be made available later, and all the participants will receive an email with a link to the recorded presentation when it's available on our website. So now, beginning with the technical questions, this is something Lindsay mentioned earlier on, and so the question is it possible for respiratory fluid droplets to evaporate in the air, leaving just the bare organisms in an aerosol?
Okay, that's a good question. I don't think that's likely. Now, we don't have any measurements of individual single aerosols to confirm with chemical composition. But the respiratory fluid contains the salts and proteins, and I don't see an easy way for the virus to separate itself from those other components of the respiratory fluid. And this is also based on my knowledge of working with engineered nanoparticles in spray products where you think, oh, because we have all these silver nanoparticles in the product, we'll see just silver nanoparticles coming out when we spray it. But no, in fact, most of what we see is are the other ingredients in the product, and we have to really hunt through it to find the silver nanoparticles, which are 100 nanometers, the same size as the virus.
Okay, thank you. There's a number of questions about heating, ventilation, and air conditioning systems. And so the type of questions have to do with, has it been found in these systems? Can they spread the virus? And any suggestion for resources or non-studies on that?
Lindsay, do you want to go or do you want me to go? Yeah.
Oh, I'll leave that for Rich.
Okay, thank you, Lindsay. So there has a recent study that I don't think has actually gone through the peer review process yet. So I believe it's still in preprint form out of the University of Oregon and the Oregon Health and Sciences University. It's a study in a hospital which has a different, oftentimes much different HVAC system than other environments. But they do show that even with significant filtration, you'll find viral RNA that gets past the filter in the system. I think the issue with mechanical systems is yes, I think that particles that contain viruses can move through the system and can get through the system. Some will be removed by the system. But if you know how mechanical systems work, especially if it's an air handling unit handling a lot of rooms, that essentially what happens along the way is that some of those particles will be removed and then they are essentially diluted from the room that the infector's in and sort of split up and diluted amongst lots of rooms. So I think the big question there becomes, even if a small fraction gets through a mechanical system, are the aerosols in the spaces that it gets to of sufficient concentration to give a high enough dose to individuals in those spaces that you would actually see a response to that dose? And that's a question I think we don't have an answer to at this point.
Thank you. And then this has much to do again with what we have been talking about. So how can ventilation and filtration really address airborne or aerosol transmission of the virus, and any suggestions for resources for people to learn more?
Lindsay, or do you want me to go again?
I think you're the, you know a lot more about ventilation filtration, so I think you should take this.
Yeah, so some of the results I showed that if you're in an under-ventilated environment, it can dramatically increase your inhalation dose to airborne particles that may contain viruses. If you're in a very well-ventilated environment, you can substantially reduce your inhalation dose to those particles. So ventilation is very important. Doing ventilation right is important. I've seen a lot of people say open windows. Sometimes you have to be cautious in doing that. If you're in an office suite and you say, I'm going to open my window to get more ventilation and the building is positively pressurized, there's going to be a lot of air moving from the office suite through your office and out the window instead of in the window. And so you're essentially bringing air from other parts of the office suite to you. So you have to make sure that ventilation is done right. But increasing ventilation in the right way can dramatically reduce your dose. And I think some of the results that I've shown indicate that using a good portable air cleaner, especially in something like a classroom that's a lot less than 1,000 square feet, may reduce your dose by 50%, your inhalation dose or more. And good filtration, that good filters in a mechanical system that are seated properly. MERV 13 filter can remove a lot of particles in the size range that Linsey talked about and I talked about that may contain viruses. So I think all of those are weapons in our attempt to reduce the dose of population to SARS CoV-2, the inhalation dose.
And any suggestion for resources for people to learn more?
So it's amazing how much information is on Twitter these days. You should definitely follow Linsey Marr on Twitter. You can follow me on Twitter. Shelley Miller at University of Colorado actively tweets great information. I think with respect to filtration, Jeff Siegel at the University of Toronto is a tremendous resource, and he tweets great information about filtration about every 3 or 4 days if people follow Jeff. It's harder when you talk about other sources of information. There's certainly a lot of information in the peer-reviewed published literature on portable air cleaners and filtration systems that are perhaps not accessible to the general public. The US EPA has a great website on indoor air quality that gives good information on portable air cleaners and other ways to reduce your exposure to all sorts of indoor pollutants. And so I encourage people to go to epa.gov and look at the indoor air quality website as kind of a starting point.
And thank you also for all this service of providing information to the public in these other informal ways. We have a question about indoors versus outdoors, and in particular whether the virus does it stay suspended indoors longer than outdoors?
Linsey, go ahead if you want.
No, it really just depends on the size of the droplets or aerosols, and then in terms of whether there's more upward and downward currents in indoors versus outdoors. I'm not sure, actually. Rich, what do you think?
So certainly air speeds are higher outdoors, and so it's possible that even larger droplets get conveyed a little bit further outdoors than indoors. Certainly the plume of aerosols is dispersed quicker outdoors than indoors, and that's why being outdoors is better than being indoors if you're near an infector. Anything that's less than 10 microns can travel pretty far indoors and very far outdoors. So I think the question is really more related to larger droplets and how far they can travel outdoors versus indoors.
Another thing I'd like to add is that there's been some nice studies showing that sunlight kills off the virus in 10 minutes, let's say, less than that in the summer, a little longer in the winter time. So if you're thinking about those kinds of time scales, then even if the virus is physically or parts of it are present, it's been inactivated or decayed to the point where it cannot cause infections anymore.
Yeah.
Great. And actually, it's a good segue on this, on several questions really on the use of ultraviolet technology in buildings. particularly would be helpful to put it in other places other than the heating, ventilation, and air conditioning system.
Linsey, do you want me to go, or do you want to go?
Yeah, why don't you take this?
Okay, so I think the question was, can UV essentially effectively inactivate SARS CoV-2 virus? Was it outside of the
outside of the HVAC systems? Yeah.
Yeah, so I mean there are upper room UV systems, and some of those are now being used. And I know of one nearby in the Northwest that uses a false ceiling and circulates the air around that false ceiling with a UV system in the ceiling. As long as you have sort of the intensity of UV is important, and if you have proper intensity and you get enough passes through the zone where the UV radiation field is, and enough contact time, you can essentially inactivate any virus with enough UV and enough time. So it's really a measure of exposure. It's sort of the intensity of the UV and the contact time with the virus.
Great. And Linsey, you were mentioning the issue of sampling earlier. So there's several questions also on air sampling, how accurate and representative it is in real environments, and is it hard to interpret the results?
Yeah, I think the sampling methods are well established, the type of samplers they use. There's questions about exactly where do you place the sampler in the room and how representative is that. Hospital rooms in general are not representative of most public spaces because the hospitals have these types of ICUs have very high air exchange rates of 12 to 15 air changes per hour. I think in residences it's more like half to 1 air change per hour. In public buildings, maybe. Rich, what do you think? The range?
So the range for, I'm sorry, Linsey.
Yeah, sure. In public buildings, what would the range of air exchange rates be?
Yeah, so if it's a public building, and if it's properly ventilated, and this is a gross generalization here, but 1 to 3 or 4 air changes per hour, depending upon the specific building. You can have lower values in residential buildings and people's homes. But again, in some schools, I've seen, unfortunately, a large fraction of classrooms sometimes severely underventilated, so you can get down to 1 air change per hour or lower in a school classroom, which is not a good environment.
Right. So if you take the hospital numbers, I could see it easily being 10 times higher. The concentrations in air easily being 10 times higher in the public space. Exactly. In terms of the sample collection, I think is standard. The analysis of number of viral RNA gene copies by PCR is very well established. So I think I have good confidence in those. The big question is whether there's culturable virus present, and there's the virus has only been shown to be culturable when you have to have really large numbers of it, and those are in clinical samples, meaning taken from patients. And it's just really hard to collect that much in air, given that the sample is often being damaged during collection.
Yeah, and I would add that I always had concerns about using hospital, samples collected in hospitals and trying to extrapolate to other environments. Hospital environments are much different in terms of environmental conditions. They're different in terms of the nature of materials that particles deposit to. They're different in terms of the amount of ventilation, the type of filtration systems, and so it's really hard to take those numbers and extrapolate other than to try to determine what the actual emission rates are from infected individuals. Beyond that, looking at the fate of viruses in a hospital and trying to extrapolate that to a restaurant or a home or a school is sort of fraught with potential uncertainties and problems.
I'd like to add that the emission rates in hospitals might actually be lower than we'd see out in the public, because by the time someone gets to the hospital and is in the ICU, they are in a more advanced disease state, and we know that the maximum virus shedding happens kind of the day before or right at the onset of symptoms.
We have questions on some of the engineering designs really that is common in the U.S. on where both the air supply and return are in the ceiling. And then the question is how much actual air exchange occurs where the people are sitting in terms of the efficiency really of this design.
Yeah. So I can say that when I mentioned ASHRAE 62.1 2019, I did say in theory. The in theory is assuming a well-mixed environment, so you're using the whole volume of the environment, and it is certainly possible that you're not using the whole volume of whatever the environment is that you're studying, and so that can, from a modeling standpoint, that can certainly cause underestimates of exposure in many cases, especially for the example that you just described, Vito, with the sort of short circuiting off of the ceiling. So, we model the best we can, and we use assumptions. I use this, what I call a screening model. It wasn't based on advanced computational fluid dynamics, but to look at those kinds of scenarios, you really need a much more advanced modeling approach. But yeah, I think the question's a good one because those things do happen in real buildings for sure.
And did you run any scenarios with office settings, particularly those with open floor plans?
So I haven't done that yet, but as a dean, I have an office suite that has about 10 or 12 people in it, and there certainly are concerns about coming back in the fall. So one of the things I'm doing is doing that. I will be doing that. I'll be modeling the office suite for the dean's office at Portland State University, and I've already purchased a number of portable air cleaners for the office suite and other office suites in our two buildings. And we'll be playing around with that and seeing how we can try to significantly reduce our potential inhalation dose if somebody comes to work, as Linsey said, that's not showing symptoms yet, but that may be quite infectious. So kind of in the process.
Yeah. And there's only so much time in your schedule, so yeah.
Thank you for acknowledging that, Vito.
I've experienced that in our interactions preparing for this, but so again, thank you. But just a few more questions before we let you go, if you have time. Both of you have talked about using masks and making some assumptions about their efficiency. Could you say something about the efficiency of different types of masks, cloth versus surgical N95, and any scenario where a face shield is sufficient?
Sure, I can speak to that. So there, N95s, of course, are rated to remove at least 95% of particles that are the hardest size to remove, which is around 0.3 microns. Filters actually remove the smaller particles even better because of Brownian motion of those smaller particles. Surgical masks can remove up to 80% if worn correctly, tightly. And then as for homemade masks, they can be all over the place. 50% efficiency is a kind of central estimate, but bandanas are fairly ineffective, but we've seen homemade masks that can easily remove 80-90% of particles of all sizes. But fit is really key, because if you have gaps, then you're short-circuiting the mask and it won't help at all. Again, masks work both ways, both for source control to reduce the amount of droplets and aerosols that the wearer releases into the air, and it also offers some protection of the wearer against breathing in droplets and aerosols from the air around them. And then face shields are good, of course, at blocking the large droplet spray, but they do not make a lot of difference for the aerosols, which can easily flow through the gaps, the gaps on the sides.
Yeah. This is a question about really trade-offs, and so imagine a central HVAC, is there a benefit to increase filtration, say, from MERV 8 to MERV 13, if it comes at the cost of reducing airflow because of the extra filter resistance?
I'll take that. I think if it's done right, if the filters are seated properly, et cetera, moving from a MERV 8 to a MERV 13 is going to give you a substantial increase in fractional removal of particles in the size ranges that Linsey and I spoke about today, and I think that increase in fractional removal is going to be far greater than any reduction in flow rate through the filter. So I think the answer is yes, and I'll point out that through a lot of modeling at my own university, we're retrofitting all of our buildings right now that we can with MERV 13 filters, moving from MERV 8 to MERV 13 for that reason. So yes.
Thank you. And the last question, I think, at this point. So, Linsey, you talk about the lower survival in medium humidity environments for the virus. What is really the mechanism for this virus inactivation?
Yeah, that's a good question. We don't know exactly. So there is this kind of U-shaped relationship between virus survival and relative humidity, where survival is good at very low humidity when the droplets are desiccated, and at very high humidity when there's very little evaporation, and it's worse at the middle. And this is something my group has been really working on to try to understand at a mechanistic level. What we've figured out so far is that at that medium humidity, you have kind of slower evaporation, which leads to concentration of the solutes and exposure of the virus to highly concentrated solutes for a longer time than if you have rapid desiccation to complete dryness at low humidities. And we think there's some interaction going on, that change in chemistry, maybe having to do with salt or protein concentrations, perhaps pH, perhaps gases that have diffused into the droplets and somehow inactivating the virus. But that's something we are working on actively.
Thank you. And unfortunately, there are a lot more questions that we will not be able to get to, including some specialized environment. And but we hope that what our presenters have shown offers principles that can be adapted to other situations. And so I want to thank you very much for all the time that you have dedicated to this presentation. It's been enormously well attended, and so again, also thank you to all those people who have participated. And at this point, we will conclude our webinar. Thank you.
Thank you.
Thank you very much.